And without further ado, then, I'll introduce our final speaker, Professor Asim Ziadi, who has very kindly agreed to do a very last-minute substitution due to the illness of Dan Anderson. Asim is a faculty in the Department of Pediatrics at Cincinnati Children's Hospital. His lab has been doing some fantastic work related to non-viral gene delivery in cystic fibrosis, and he's going to share some of their insights in terms of the challenges and solutions related to delivery through animal models and into humans.
Thanks, Millie. So please bear with me as I stumble through this. When Millie says this was a last-minute kind of thing, she literally means that. I prepared this in the hour before the session. So, but we thought we'd just give you something to look at rather than not. So what I wanted to present to you is the problem that we think about all the time, which is in my lab, which is gene transfer for cystic fibrosis and how we can get it to work in vivo, which CF, the disease that we're interested in, sets a bunch of definitions up for us that we have to abide by and work within that impose a bunch of limitations on us, and I'll walk you through those. So the history of gene therapy in CF is pretty long. The gene was sequenced in 1989, and very soon after that was done, people ran to the lab to try to do retroviral gene delivery for CF, adenoviral gene delivery for CF, AAV. And so viruses have been tried, liposomal vectors have been tried, and every vector that's been tried, and I'll just go through all of them here, has run into issues. So Kevin, mused about the differences between viral and non-viral vectors. In CF, that has been much highlighted. So the viral vectors in CF have trouble accessing the cells from the lumen, and so adenovirus, for instance, has its receptor, and most of the adeno-associated virus serotypes have their receptor on the basolateral side, so you have to have injury for these to work very well in the airway. There's packaging limitations of AAV because the gene for CFTR is pretty large. The lipids ran into problems with non-dividing cells, and inflammatory responses by any vector that anybody used, and these were reported in CF for adenovirus and lipids, were a big problem because CF is an inflammation-causing disease where people present with severe lung inflammation, and it's not something you want to exacerbate. So we have been trying to focus on a strategy that's safe and can access these cells. In addition, CF is a multi-organ disease, so it affects all the organs in the body. The liver, for example, is the second most affected organ in CF. It's hardly ever talked about, but many people suffer from severe liver disease in CF, and it's potentially a gene transfer, gene delivery organ target for CF. The issue in the lung where we have been focusing for gene delivery is even if you get excellent gene delivery and gene transfer that lasts for the life of most of the epithelial cells that are important in CF, you still have to deal with the turnover of the cells, so we will have to re-administer these particles or whatever vector is used. And that becomes a limitation that is a very important one, since you have to do gene transfer for the life of the patient. Unless you can, of course, target stem cells, which is challenging, and these have been very hard to find, stem cells for respiratory epithelia and ciliated epithelia in the airway, which are the most affected cells in the CF airway. So to be able to succeed in gene transfer for CF, you have to have a vector that can package the cDNA for CFTR, which is about 4,400 bases. Quite large. There's been clever approaches of using AAVs to even do this, where you can use the concatemerization quality of AAV and do two halves of CFTR and have the AAVs concatemerize in the target cell. This hasn't always been an absolute limit, but it definitely does help to have a large packaging capacity. And you do need to get about 25% of the cells in the airway and about 10% of normal endogenous levels of mRNA of CFTR to actually correct the airway and the fluid transport across the airway to be able to get a full correction of the disease. You have to sustain it for years, so we have to do repeat administration, and that of course becomes limiting with vectors that induce immunogenic responses. And vectors must be safe and not toxic because of the long-term therapy and the virtue of the disease of already existing toxicity and inflammation in the patients. So an extended duration obviously is desirable as it is for any other disease that we try to target. So we've been able to minimize let me just skip through this. Actually, that's not really that important here. In addition to some of these limitations and important characteristics of a vector that you'd use in CF, you have to contend with what the airway presents as a challenge. And so this is an example taken from Bridget Hogan's review of the airway and the cells that are in the airway, and no less than about 40 different cell types in the lung. But along the airway, you really want to target ciliated epithelia and then serous cells of the submucosal gland, which you can't really actually see here in this depiction, out of all these other cells that are present, like type 1, type 2 cells, or macrophages in the alveolus, or mucus-producing cells in the airway. And so targeting is an issue, getting the right cell. To get to these cells, if you're coming from the lumen, if you're coming from the basolateral side, you have to go across blood vessels and then the basement membrane, and then approach these cells from a basolateral side. Most of the time, things that get into these cells come in through the airway. There's injury here somewhere, and they manage to get around these cells, and that's how they get to the basolateral side of these cells. But you also have to contend with a layer of mucus floating above this layer of fluid called the airway surface liquid. And this mucus is actually not as thick as you could imagine, we all imagine mucus to be. It's actually 99% fluid. But it can slow down particles, and so particles will actually slow down as they move through this layer, and mostly get trapped into this layer of glycocalyx over here, which is a network of glycosyl groups decorating the surface of epithelial cells. So there's a severe challenge in actually getting into the target cell. In tissue culture, this is a model that we use here of air-liquid interface grown cells, and basically you use these filters where the cells can sit on these semi-permeable supports. We get these cells from patients from explants. So we're able to test these primary cells. They become confluent, form very high resistances, they become ciliated. There's mucus cells, clara cells, ciliated epithelia, respiratory epithelia that are in these mixes of cells that we grow. And they're actually quite good models of electrophysiology for CF at least, and has been one of the models that we've gone after. It leaves something to be desired, and this is what they look like in culture for us. You can see the cells side by side here with the cilia on top of each other. We can talk about the limitations of this model, but that model allows us to do high screening before we go in the animals. The major issue, aside from accessing these cells for us, which a lot of people have studied, so there's a number of labs that have focused on what happens out here. I talked to you about the layer of airway surface liquid with the mucus on top of it. There's a number of labs. For example, Justin Haynes at Hopkins has been looking at how to get particles through mucus more efficiently and then to the cells. My lab is focused on what happens inside the cell, so basically what the traps inside the cell for particles are. And we think if you sort of look at the most basic experiment where you take the immune system out of question, you take the mucus even by washing the cells out of the question, the efficiency of nanoparticles compared to viral vectors is about a millionfold less efficient in transfecting these cells. And so there's got to be other traps within cells that are important and they're worth paying attention to. The particle that we use is similar to the one that Kevin talked about in his talk, which is a 30-mer of polylysine that we pegylate, and we use either 5K peg or 10K peg that can then be complexed with DNA most of the time, but we've also done RNA. But again, we have an interest in long-term expression, so DNA for us, and not having to re-administer every couple of hours, and so DNA for us is the mainstay. There's advantages of this particle over other ones that we've tried over the years, and we can discuss this. There's been about 30 papers associated with all this work. I'm sort of going through really quick here, and we can talk about the virtues of this latest peptide that we use. But it produces particles that are pretty homogeneous. This is probably not true in most cases. It's probably up to 7 particles, 7 DNA molecules per particle, and not just a single one, so I apologize for having that still in there. But we can concentrate DNA up to 12 milligrams per mil, and the stability is very high. So you can put this in a refrigerator for 2 years, then go grab it and compare it to something that you just made, or compare it to what it did when you first made it in vivo, and the efficacy is the same. The particles look like this on EM. So these are actually tagged particles, so they have gold tags on them, which is what you see here in very dark contrast. These gold tags are then developed with silver, so you can actually see the particles once you put them in cells or in vivo. So we do this because it becomes really difficult to see these particles here, which are the rod-like hairs that you see here. That's our particles. That's what our particles look like. They're not spherical like Kevin's particles are. And so we produce these rod particles, but they're very difficult to see in vitro in EM or in vivo in EM unless you have this big dark contrast tag on them. And so we do that so we can actually see, do our particles get across this extracellular barrier here of the glycocalyx and the airway surface liquid and the mucus, which you can sort of see a line here where if you blew this up, you would see all of the particles. Many of them are stuck here, but particles are also descending upon the cells, and I'm going to blow them up for you. But that's how high let me just go back here. This is how high the layer of fluid on top of the cells is, and these are the cilia here beating, and these are 3 different cells here right next to each other, and you can see a nucleus here. And so if you blow up the cells, this is the same EM, just with a higher magnification. Now look at the cilium of one of these cells. So here's a cell. This is the apical surface right here. You can see the cilia. And you can start to see the particles coming down along. This is, I think, about an hour after we added them, coming along down the cilia, some of them associating, some already associating with the apical surface of the cells. You can see it a little bit better here. But you can see that just looking at what the particles would look like in cells, it would be very difficult to actually tell that these are particles. So it's good that we have these tags on them. It helps us sort of follow them around. And in about 2 hours, we can start to see some internalization. I'm sorry, I can't see it too well. Yeah, of these particles. You can see them at the surface here. We've even captured them entering the cell. And so there's internalization taking place here, and you can see the particle hanging off of these 3 tags here. So we can get inside of these cells. There's just an example of tagged particle inside of a cell. Sorry. We can get inside of these epithelial cells in culture, these primary cells from patients, but we can also do it in vivo, and this is the result of, there's many publications out there on us getting into the lung with BLI, and these are typically what we see for the controls. And we get into the epithelial cells that we really desire, although the expression level is, expression is always patchy, and this is a problem that we've actually tried to address, but it seems like is more related to cell cycle than anything, and that's why you get patches of cells like this across an airway rather than a dispersed set of cells. But there are examples where we see dispersed expression. And also we get type 1 and 2 cells that are actually not in large airways. Toxicity-wise, I'm going to skip over this. This is just an example here. I probably should blow this up where you can see like these 2 cells have expression while the ones right next to them don't. I like that immunohistochemistry because this is impossible to fabricate, and we just don't know what's going on with these 2 cells versus the 2 cells right next to them. But there are examples where we get very, very extensive gene expression, and this is just a ratio here of animals in one experiment that we did where 3 out of 8 got this level of expression. The toxicity on the particles is not significantly different from just dosing mice with saline alone. Even when you use a high dose of DNA, 100 micrograms down into the lung of the mice, as compared to using a low dose of genomic DNA that's not compacted. And of course, we use a CpG depleted plasmid, and so we're really comparing CpG depleted plasmid here to E. coli bacterial DNA. And when you look at the cytokine measurements for inflammatory cytokines, we really don't see any significant increases in the inflammatory cytokines, with the exception of KC and IL-6, which are elevated in mice about 24 and 48 hours after we do the gene transfer. So they're pretty safe. We moved the particles onto a trial in humans, and this is one of 2 trials that's ever been done in CF with nanoparticles. One was done in the UK about 3 years ago. This was done in Cleveland a number of years ago, more than 3 years ago, almost 12 years ago now, where they used the nose as a model because the nose has respiratory epithelia that you can brush the particles on. And you can then look at these respiratory epithelia, and there'll be a reflection of what would happen in the lung, because the respiratory epithelia and the nasal respiratory epithelia can be used to model what happens in airway, lower airway respiratory epithelia. And this was mostly a safety study, but I'll show you the efficacy data in a second. It was a small study; only 12 subjects were studied, the responses as far as inflammation or chemistry changes or hematological changes or changes in complement activation were really zero. No evidence of inflammation in the nasal washes that we got from these cells, and nothing systemically evident. There was one patient that developed an increase in IL-6 in the serum, but they later developed an upper respiratory infection, and so it was not due to the vector. Efficacy-wise, the data was actually pretty promising. So these are the 3 cohorts here of the dosings that we gave to the, that were given in the study done by Constant. We were collaborators with Mike Constant and Pam Davis on this. But the dosing was 2 patients got 0.8 milligrams in the nose. This is in terms of DNA, 2.6 and 8 milligrams. And in the highest dose group, we saw some patients, this is now a function measurement of chloride transport in the nose called a nasal PD measurement, and we can talk about how you do this if you'd like. But basically, deflections downwards in this, or a decrease in millivolts, it signifies secretion of chloride through CFTR. It's done. The assay is done with a Forskolin stimulation of CFTR after you deplete the chloride in the electrode that you're measuring with. And so you can see that some of the subjects entered the normal range, some trended towards the normal range, almost all trended towards the normal range, and then they returned back to CF levels after 13 days. We designed the study this way. We used a CMV promoter so the expression wouldn't last because we wanted to see that we didn't damage the noses in any way to get this artifactual measurement over time. And so we were able to show that. But the efficacy, so some of the efficacy data was was split in my mind. At the high dose in the trial, 6 out of 6 patients had DNA on both sides, so we had actually dosed one nose and tried to use the other nostril, one nostril, and tried to use the undosed nostril as a control, but it turned out that the DNA moved from one nostril to the next. But by 14 days, 5 of those individuals still had DNA in there, so and they were only dosed at day 0. So the DNA was protected. This was we did southerns on this. We did PCR on this, and we PCR'd long pieces of DNA, so not the short pieces. So the DNA was actually not degraded and protected in these folks for up to 14 days. That was somewhat promising, but the most discouraging thing about the study is we couldn't detect any mRNA levels for CFTR. The nasal potential measurement, difference measurements I showed you, the high dose group was very promising, and certainly there was trends in the lower dose groups that then returned to normal and were different from baseline. So the clinical trial showed some benefit and good safety, but the efficacy remains as an issue, I think, and it's something that we've gone after. There's different important issues to go after. Some folks, including Kevin alluded to this, and Ewan before him, will go and make different kinds of vectors. We sort of did that. We played around with the length of the polylysines. We tried polyarginine. We tried PER. back in the day, but I got sort of turned off on that very quickly. And, I thought that there were better people that could do it better than us out there, just mass produce different kinds of vectors. I should say it's my opinion that despite of about 35 or 40 years of doing that, because these vectors have been used for a very long time, just making different kinds of vectors, we still have vectors that don't beat the efficiency of some of the original ones, and they're much more toxic. So, the angle that we took, aside from decorating the particle, which is something I won't talk to you about today, but we do put targeting ligands on the particle, is to look at these intracellular barriers to gene therapy, and we started looking at the particle biology. So, in looking at what the particles actually interacted with, you can sort of see here, actually I can't see it, so yeah, here, the processes associated with the proteins that interact with proteins that interact with the particle after they're added to these primary cells. And you can see that the top 3 or 4, actually all of them make sense. So cytoskeletal class proteins, translational proteins was a little bit troubling for us because we were interested in DNA, and of course getting DNA stuck in the ribosomes is not a good thing. But this might be a good thing for RNA. So I think there are traps in the cells for nucleic acids, and you have to avoid some of these traps if you're doing DNA and enhance them for enhance them for RNA, and vice versa. So just as a quick example of a couple of these interactors, one of them that we found and published on was a glucocorticoid receptor, which we found did interact with the particle. We identified that by mass spectrometry and found that if you then use cortisone based on that information to bring more glucocorticoid receptor up to the surface of cells at the right time and at the right dose, you can enhance gene transfer by threefold by just giving it at the time of gene delivery, not really keeping it on all the time. It's just at the time of gene delivery. And we've built this now interactome here for the particle that actually Steve Rayner is going to be giving, is a postdoc in the lab that's going to be giving a very nice poster on this, poster 592, where he's going to have more information on this. This is just what I could piece together so last minute. But we find interaction with spectrin, with APC1, glucocorticoid and nucleolin, and HSP90 also interact with the particles. This interaction with nucleolin here is affected heavily by what's going on with HSP90 and glucocorticoid receptor regulation of nucleolin levels at the surface of the cells. And we can modulate these things with drugs like cortisone or gadenomycin or 17-AAG, as well as knock out these proteins, for example, if we want to do stuff ex vivo. So this just is an example of, and I think Steve will have pharmacology data at his poster, which I think is even more promising than the silencing of spectrin or APC1. But you can see that we're talking about tenfold, 1 or 2 orders of magnitude of increase of gene delivery by just manipulating the biology of the particle. So I think, in summary, the particles have a long shelf life. They're easy to make. The materials are synthesized. We don't have as much trouble with the chemistry. It's pretty straightforward to us. We use maleimide chemistry in most of our conjugations, and we make sure these are made to GMP standards. The particles are safe in vivo. They mediate gene delivery in mice and in humans. I think retargeting the particle is possible, and I didn't show you the data, but we actually can change the surface, decorate the surfaces of these particles and not really change their characteristics drastically as far as turbidity, important characteristics as far as turbidity and sedimentation or immunogenicity if you use a low amount of peptide. Of course, the targeting will depend on what application you want to use. And I think studying the determinants, the biology of the particle, which I see an AAV biology session at the meeting, but I have never seen a nanoparticle biology session at this meeting. And sort of Kevin came up here and gave his opinions, which I actually admire that he did that. That would be my opinion of the day, is we should start thinking about the biology of these nanoparticles instead of just making thousands of them all the time. Although I think the high throughput stuff will work. I'm a believer in high throughput. I just think, intelligent design, if I can use that word, is probably something that we could probably do if we just slowed down and thought about the biology like the viral people have for 50 years. So I think just to acknowledge everybody. Sorry if I went over, folks, but so Steve is actually going to give a poster 592. You can get all the details that I didn't give you about this most recent interactome stuff. Matt is a very talented research assistant in the lab that has helped with these studies. Dana's another postdoc in the lab, and Sombai and Keishel were previous members in the lab that helped with this project. And then the collaborators are Pam Davis, Michael Constant. They did the clinical trial. And I trained in Pam's lab, by the way. And then Shenghong Li for the imaging studies, Elizabeth Wright at Emory for the EM studies, and J.P. Clancy at Children's for a number of other studies that I actually didn't get into, but we're doing some clinical studies now at Children's, and J.P. is part of those. And there's the funding, so thanks.
All right, so time for questions. And I'll lead off with a quick one while people are figuring it out. Wanted to ask more about the interactome analyses, and in particular, I wondered if you could just share a little bit more about your approach. I know often there are many potentially really interesting hits, and I'm wondering how you're going about sort of identifying those that are the most important ultimately to the transfer of the.
So we used two approaches. One was an antibody against the PEG, which you can, like was mentioned actually during Kevin's questions, they're available. And so we used a pull-down approach and also a fractionation approach, and compared those to just saline as a control to see, what interacted with the particle and what didn't, with a mass spec, identified with a mass spec. And we then went into cell lines and knocked every one of these out, which took a very long time. I didn't show you any of that, but there's about 530 proteins that came out of this analysis, and we did really high throughput knockdown of all of these proteins to see if it affected delivery in those cells, in HeLa cells. So it's not, it's not something I would publish, but it's something that helped us pick the ones that were really important, and we settled on the ones that were really important and studied those in the primary cells in the animals.
I know we're short on time. I think we may have one question. All right. So why don't we thank all three speakers one more time for a fantastic session.